Good afternoon, everybody. Thank you so much for joining us in Medical Grand Rounds today. It is absolutely my greatest honor to present my former mentor, and he's really kind of currently my mentor still, Dr. Raymond Chung from Massachusetts General Hospital. He is the director of hepatology and the director of the Liver Center at Mass General, and he's a professor of medicine at Harvard University. He's also the current president of the American Association of the Study of Liver Diseases. Ray's been an absolutely prolific physician scientist since he graduated from Yale School of Medicine in 1986. He completed his residency at Hopkins, and then he went for GI fellowship at MGH and stayed on as faculty starting in 1993. So he's been there a long time, and during that time, his research has been focused on understanding the pathogenesis and treatment for viral hepatitis primarily, especially hepatitis B and C, but his range of research investigation has really covered a broad number of other liver diseases. To the end, he has authored over 350 scientific manuscripts. He has held over 46 sources of funding, including numerous NIH R01s, U's, T32s, as well as several industry-sponsored grants. He has mentored over 115 trainees during his time, most of whom have gone on to their own scientific careers in both academia and industry, and I'm but one of those. So the number of national and international accolades that Ray's received over the years are really just too numerous for me to name here, but he's been the recipient of numerous Harvard Teaching Awards as well as Clinical Investigator Awards, and he's currently the Kevin and Polly Maroney MGH Research Scholar. On a personal note, I've been very grateful to have Ray as a mentee, as a mentor. I've known him since I was a medical student applying for residency, and he's really ushered me through my scientific career during my residency and fellowship. To me, he represents the sort of pinnacle of what a dedicated physician scientist can accomplish when they set their mind to their efforts and have tangible goals, both for themselves and their trainees. So I try to be as much as I can like him, and so I was so happy that he accepted my invitation to give this talk to us today. So please help me in welcoming Dr. Chung, and he'll be talking to us about new prospects for Hep B. Can we now visualize cure? Ray?
Annie, thank you so much for that entirely unnecessary introduction. It was over the top, but I think the singular most important sentiment I have is one of just pride and pride in mentorship. That's perhaps the single most important thing that you know has really fulfilled me most in my career. And there are no better or shining examples of that than Dr. Kruger herself. It's great to see old friends again, and actually to share with you essentially a topic that has really flown under the radar screen for years, really eclipsed by hepatitis C. By the way, can you see that on the screen? Is the presentation there on the screen? Okay, great, terrific. And really deserves new attention in the face of the success we've seen with hepatitis C related cure. So we're gonna focus on Hep B and talk about prospects for its cure. These are my disclosures. And just to frame the talk, we'll start with a consideration of the clinical problem, our understanding of chronic hepatitis B and its sequelae, and to consider cure, we'll have to consider, of course, the virology of hepatitis B and its pathogenesis, and more specifically, the virus life cycle and specific targets. And then we'll move into the meat of the matter, which is really the new prospects, direct-acting antivirals and even immunologically targeted treatments for hepatitis B in our effort to bring about cure to this chronic infection. So by way of inspiration, I think it's worth again noting that the road to cure of the highly related hepatitis C virus, highly related in that it causes a chronic infection and disease of the liver, really I think has been an extraordinary paradigm for the success of science and discovery, leading to in this case full cure of this RNA virus, and of course hepatitis C virus, and of course recently awarded Nobel prizes to Professors Alter, Houghton, and Rice. And really, when we look at the playbook for the success in hepatitis C, it was really exploiting perhaps the Achilles heel of that virus. So by way of just a reminder, Hep C is an RNA virus with no DNA intermediate, so it has no latent form in the nucleus. So it is therefore dependent on continuous replication in the cytoplasm. And so with a multi-target, multi-mechanism of action, direct-acting antiviral strategy, it is theoretically possible then to extinguish that life cycle by putting enough antiviral pressure on without allowing emergence of resistant variants. And that's indeed what happened with multiple classes of therapy being applied successfully to Hep C. So the question then is really realistically, can we think about and pivot all of our attention and resources to that other chronic viral infection of the liver, hepatitis B virus? And the answer I will share with you over the next several slides is, I think, a qualified yes. But before we do that, let's consider the scope of hepatitis B disease, and that is, pardon me here, shown here. And this is the scope of the problem. Now, there are over 250 million persons chronically infected with hepatitis B. And by infected, I mean chronic hepatitis B surface antigen positive, the sine qua non of hepatitis B chronic infection. It is, it remains the leading cause of hepatocellular carcinoma worldwide, and still contributes to nearly 1 million deaths annually, according to the World Health Organization. This has been particularly frustrating because we, of course, know that hepatitis B is vaccine preventable, yet its prevalence has not budged over many years, remaining largely static, and that's because of the lack of penetration of vaccines to where the disease resides. And it resides in these hotspots, as shown here, particularly in red, where seroprevalence rates exceed 8%, such as East Asia, Southeast Asia, and Sub-Saharan Africa, to name several sites. So with all of the flurry around the cure of hepatitis C, the World Health Organization decided that it would lay down the gauntlet, that by the year 2030, we should witness elimination, or essentially elimination, of hepatitis C and hepatitis B, in which incidence of new infections of hepatitis B, in this case, would be decreased by 90%, and corresponding liver-related mortality by 2/3. And this, of course, for hepatitis B would be accomplished through maximization of screening and, of course, the administration of universal birth dose vaccine, because the perpetuation of the epidemic occurs with mother-to-child or vertical transmission. But for those 250 million persons who have chronic infection, that will, of course, not suffice. We need to look at curative therapy for those who have chronic liver disease. And so with that, I think we're going to need to deep dive into hepatitis B and our understanding of this fairly complicated virus. So this is the molecular architecture of hepatitis B virus as we understand it in 2021. It is a small, partially double-stranded DNA virus, very different architecturally from hepatitis C virus. It's a DNA virus with multiple open reading frames that are indicated in this cartoon, with 4 major gene products, including a surface envelope glycoprotein, the HBV DNA polymerase, which has a reverse transcriptase activity, the pre-core core protein, which encodes the HBV core capsid protein, as well as an embedded protein here known as E antigen within the pre-core region that is synonymous with active viral replication. And then the aptly named X protein, which is essential for the viral life cycle, but whose mechanisms of action still remain to be fully clarified. Within the pre-core region, this E antigen encoding region, lies a mutant known as a pre-core mutation that leads to premature termination of the E antigen and can lead to a chronic state of E antigen negative chronic disease, which is every bit as insidious as E antigen positive disease. So again, as we think about the ability to successfully cure this infection, we're going to need to understand its life cycle. much as we did with hepatitis C virus. And this is a fairly complicated life cycle, and I want to walk you through this. So hepatitis B, as a virus, enters through, it enters the hepatocyte in this case, through a receptor known as the sodium taurocholate co-transporting peptide, or NTCP. It's a bile salt transport protein. The HBV then becomes internalized through receptor-mediated endocytosis, and then that membrane becomes dissolved, leading to release of the virus and that the movement of the genetic material, the HBV double-stranded DNA genome, into the nucleus, where it takes up stable shop as a highly topologically stable form known as covalent closed circular, or CCC-DNA. This exists as kind of a mini chromosome outside of the nucleus or outside of the chromosome, and it acts as the template for transcription of variable length RNAs, including a long pregenomic RNA, which acts as the template for subsequent viral protein synthesis. And that protein synthesis, of course, includes the reverse transcriptase polymerase function, which reverse transcribes that RNA into a first DNA strand and then into a second DNA strand to complete the double-stranded DNA genome. There is also a core protein being generated, which encapsulates the HBV genomic material. And then that becomes, that incorporates these surface envelope proteins into mature virions, which depart the cell through secretion, only to infect new target cells. At the same time, there are smaller length RNAs being transcribed, again, from that CCC-DNA, and they are, in fact, freighted for a different fate. And this is to be translated into surface proteins of variable length, so-called envelope proteins, which can then be secreted alone, isolated as so-called subviral particles, otherwise known as surface antigen. So somebody may not, it's possible for one not to have active replication, yet to have continued surface antigen synthesis and secretion. And we call this the inactive carrier state, in which the surface antigen is being secreted, yet there's little to no active viral replication taking place. Now, it should be noted that the CCC-DNA is actively replenished because any unincorporated DNA that doesn't go into virus finds its way through a very efficient recycling step where that DNA moves its way back into the nucleus to replenish stores of CCC-DNA, which again makes this an extraordinarily difficult species to clear. It should also be noted that HBV DNA sequences can also be integrated into the host chromosome itself, further complicating the situation in terms of alternate sources of viral protein synthesis. So when we think about chronic liver disease related to hepatitis B, we think of this as being immunologically mediated, and we'll cover more of this later as we talk about immune-mediated therapies. But this is principally orchestrated through the CD8 positive cytolytic T cell, which ordinarily would bring about clearance of virally infected cells through their presentation of viral peptides with class 1 HLA, and the clearance through receptor through programmed cell death or apoptosis. But with the failure to clear, such as occurs in chronic infection, there's a smoldering picture of cytokine release by both CD8 positive and CD4 positive T cells, resulting in the cytokines listed here, which in turn both directly activate hepatic stellate cells, which are essentially in health retinoid storage depots, but become, once activated, essentially fibrosis or collagen producing cells, which leads again to the phenotype of fibrosis. And this can be abetted by activation of macrophages in the liver of Kupffer cells, which in turn release TGF-beta to further promote fibrogenesis and fibrosis. And so this is what occurs with chronic liver disease once we have failed to clear hepatitis B. And this natural history plays itself out over many decades, in this case 30 to 50 years, which ordinarily would seem like a long time, but for most people in the world who've been vertically infected and infected from birth, this can lead to premature morbidity and mortality, and in the form, of course, of permanent fibrosis and cirrhosis, and the development of hepatic decompensation, liver failure, and an independent risk for hepatocellular carcinoma or liver cancer, and an increased risk, of course, for mortality. So this is the nemesis, the chronic progression of fibrotic liver disease eventuating in hard clinical outcomes. So when we talk about cure, we are looking at a tall order here because HBV is a lifelong infection with high latency and a highly stable form in cccDNA. So true virologic cure of hepatitis B will be very elusive, unlike the situation with hepatitis C. Yet we can still think about endpoints that are desirable clinically. And one of those is what we call functional cure. And these are individuals who are able to maintain absence of replication, namely undetectable HBV DNA, off of any form of treatment. And most importantly, who lose hepatitis B surface antigen with or without acquisition of a neutralizing antibody to hepatitis B surface antigen or anti-HBs. This is clinically relevant, and I think it's important to point out is naturally achieved in infection later in life, and more on that in just a moment. I think far more elusive will be the idea of complete cure, sterilizing cure as it's sometimes called, in which cccDNA is actually fully eliminated. This is a considerably taller order, but a functional cure is something that we think is graspable. So I mentioned to you earlier that the vertical transmission leads to a high rate of chronicity. Very high, in fact. I mean, so in this chart, we can see here that 90% of individuals who are exposed in the perinatal period go on to chronic infection. And that's owing to an immature immune response and essentially a state of tolerance to hepatitis B. With the older age at the time of exposure to hepatitis B, there can be a more robust and mature immune response, such that not only do we see an inverse in the rate of chronicity in older children and adults when they first see Hep B, in which the chronicity rate is now not 90%, but 10%. And not surprisingly, there is also the development of a higher frequency of symptomatic infection, i.e. Icteric infection, leading to, again, a brisk immune response and clearance of hepatitis B. So the notion here then is that there is the possibility of functional cure to be achieved. Now, again, in contrast to the vertical transmission, which looks something like this, the typical movement to chronic HBV in which surface antigen persists, E antigen is present for years, be consistent with active replication. IgM appears early to antibody to hepatitis B core, and then it is eventually supplanted by IgG subclass, which persists indefinitely. These individuals never mount anti-HBs and have a state of chronic infection. They may seroconvert ultimately to anti-HBe and may develop E antigen negative chronic hepatitis B. Now, in those persons who are exposed in adulthood, this is the scenario that prevails, in which a surface antigen is lost, the IgM comes and goes, but eventually is replaced by antibody to HBs. This neutralizing antibody is protective against future infection. And there is quick, rapid loss of E antigen and an anti-HBe seroconversion. This is the picture of functional cure and is accomplished in natural infection that occurs during adulthood, for instance. So more on this later when we talk about how do we get to functional cure. So when we think about antiviral strategies against hepatitis B, then it's important for us to revisit the steps involved in replication in the viral life cycle itself. And the focus, really the monochromatic focus has been on this single target alone. And this is, of course, that HBV polymerase, that reverse transcriptase step, and to which all small molecules have been developed thus far. And these, of course, are the nucleoside analogs approved here by the FDA over the years, lamivudine and dafivir, and the two current standards of care, entecavir and tenofovir, and its prodrug tenofovir alafenamide, along with another nuke called tobivudine. And the outlier here would be PEG interferon alpha 2A, not a nuke, but a antiviral cytokine, which has very little uptake clinically because of its significant adverse event profile. So essentially, we have been relegated to a single target strategy over many years for hepatitis B. Now, that has been a very successful single target strategy insofar as Nukes have been very successful in suppressing HBV DNA to undetectability, such as is shown in this slide that takes our current standards of care, entecavir and tenofovir, given as once daily meds, producing upwards of 90 to 100% rates of undetectability in e-antigen negative chronic hepatitis B, as an example, in contrast to interferon, which produces only about 60% rates of undetectability. Yet, the problem is that while we can take the horse to water, we cannot make it drink. And so excellent viral suppression results with these patients, but as you can see here on this y-axis, fewer than 4% of patients ever lose surface antigen at 1 year of therapy, and the numbers aren't much better than that as you go into subsequent years of treatment. So essentially, we are unable with monotherapy with nukes to get patients effectively to functional cure. But let's be sure that functional cure, and I want to emphasize this, that functional cure is associated with even greater clinical benefits. So this is a study from Southeast Asia, from Hong Kong specifically, of over 20,000 patients who were on nuke suppression for many years, with a median follow-up of 5 years. And as you look at these patients in terms of their subsequent risk for hepatocellular carcinoma shown on the y-axis, you'll see here that those persons who had incomplete viral suppression did the least well, i.e. had the steeper slope, than those who had complete viral suppression, i.e. undetectable HBV DNA. But if you take the very small number of patients, who numbered just somewhere over 100 of these patients, who experienced surface antigen loss, i.e. functional cures among this group, their rate of HCC subsequent development was substantially lower. So there appear to be clinical benefits that in addition to those seen with suppression alone in persons who do get to functional cure. So we do think that there are additional benefits clinically to be gained with functional cure, not just simply being able to come off medications. So again, as we think about the approaches to functional cure, in keeping with our understanding of the virus and its many life cycle steps, there could be direct antiviral approaches that work on those life cycle steps, but there can also be steps that work on the host immune response to enhance clearance of virally infected cells. So let's consider those in sequence, starting with virological approaches, or direct acting antivirals, if you will. And we'll consider polymerase inhibitors, they're the standard of care, entry blockers, nucleases to edit and degrade cccDNA, RNAi strategies to essentially block viral protein synthesis, HBV capsid or core assembly modulators, and then inhibitors of hepatitis B surface antigen release. So in sequence, we'll consider now, you've heard already about the polymerase inhibitors. Let's consider entry inhibitors. Is there a way we can think about just blocking access of the virus to the target cell in the first place? And the good news is that we're now starting to see candidates emerge in this space. So as I said, HBV and its actually sister virus, hepatitis delta, which is a defective viral particle that relies entirely on HBV for its viral life cycle, enters the hepatocyte through this receptor known as the NTCP molecule. It's a biosol transporter, and theoretically would halt the development and accumulation of cccDNA in the first place if it were efficiently blocked. There is such an agent that blocks NTCP, that binds tightly to NTCP, known as Bulevirtide, formerly known as Myrcludex B, that has a potential utility in antiviral prophylaxis, but certainly as part of a combination antiviral regimen. Now, the theoretical limitation here is because it's a host biosol transport protein, is that it inhibits biosol transport and could lead potentially to some cholestatic consequences in theory. So these are exciting data from Bulevirtide. And this was, I'll walk you through this. The Bulevirtide was given in a variety of different forms, either as monotherapy, shown here, or PEG monotherapy as a comparator, PEG interferon, or in conjunction and combination between Myrcludex B and PEG interferon. And what you will see here is that 2 different doses of Myrcludex B were used with PEG interferon, and it's those combination arms compared to the monotherapy arms that actually brought about substantial reductions. In this case, this was a delta Hep B trial, resulted in substantial reductions in delta RNA to undetectable in a substantial portion of patients. Half of these patients went to undetectability with the combination therapy, as opposed to only 4 of 30 in those who received monotherapy. The fantastic news is that after 48 weeks of treatment, and in the follow-up period, the 24-week follow-up period that followed, that sustained loss among those 15 who achieved undetectability was seen in 12 of those patients. And this is the first time we're really seeing substantial sustained loss of delta with any therapeutic strategy, because delta has been extraordinarily difficult to achieve sustained clearance in. Moreover, there was improvement in biochemistry shown here in terms of ALT levels improving, and even for hepatitis B, and looking at surface antigen levels, there was in nearly a third of patients who received combination therapy an over 1 log reduction in surface antigen, and in each of those individuals who experienced that reduction, that was a sustained reduction of surface antigen. So there appears to be a synergistic effect of PEG interferon in conjunction with Bulevirtide on both hepatitis delta and hepatitis B surface antigen readouts, and really an intriguing curative potential for both delta and possibly B using such a strategy. This was well tolerated. We talked about cholestasis. There was an asymptomatic elevation of bile salts. There was no impact on alkaline phosphatase or other markers of cholestasis. So a promising approach indeed. So what about moving further down the life cycle to nucleases and trying to manipulate cccDNA itself to lead to potentially its degradation, it being the foundation for everything that follows? So this has not made it into patients yet, but I just want to share with you a strategy using another recently Nobel Prize awarded technique, CRISPR-Cas. This is, of course, is editing of gene sequences. In this case, a CRISPR strategy was applied to a stable lentivirally transfected cell line that harbored hepatitis B surface antigen as a constitutively produced product. In this case, the introduction of a guide RNA against surface antigen was introduced that brings about a termination codon here from CAG to TAG within that cell line, inducing failure to produce surface antigen shown here on this graph. So the proof of concept is demonstrable here that CRISPR editing can bring about a shutdown in this case, HBV protein synthesis. So thinking about applying this to patients moving forward, the main challenges for this approach will really be delivery to the nucleus of the hepatocyte and really, plenty of nuclei in this case, given the penetration of cccDNA in many hepatocytes, as well as the concern for any possible off-target effects of this gene sequence approach. The next approach would be to take the transcripts that emerge from the nucleus and see if those can be selected specifically in a sequence-specific manner for inhibition and degradation and the inability to produce protein from those templates. And this is, of course, a strategy known as RNA inhibition or RNAi, or small interfering RNAs as shown here. And what they effectively do is once delivered, in this case through a lipid nanoparticle, they're taken up by endosomes and then released from endosomes and then bind those RNAs, target RNAs I just shared with you from HBV itself, to recognize those sequences and form what's known as an RNA-induced silencing complex that brings about the selective degradation of that, as it were, messenger RNA. And so a number of hepatocyte-targeted ligands like N-acetyl-galactosamine or GalNAc have led to a brisk uptake of this payload into hepatocytes, where again, it's taken up and released into the cytoplasm to bind those HBV-specific targets. And the premise here is to block surface antigen in particular, and possibly other viral proteins such as polymerase and core. And the notion here is that surface antigen produced in excess, remember that it is secreted as a surface antigen, may be acting as an immune decoy and actually suppressing or subverting the host immune response. So the reasoning might be that not only would the knockdown of pregenomic RNA block replication by depleting availability of proteins for replication, but also could liberate the immune response to Hep B itself. So there may be a twofer here in terms of the ability of these RNAi approaches to work on both immune responses and on viral replication itself. And so RNAis have been developed, such as with this particular agent called ARC520, to a number of different viral sequences as shown here in this cartoon. And so data from that very same molecule, now known as J&J 3989, given as 3 doses given 4 weeks apart, shown here, resulted in essentially sort of 2 kinds of buckets of patients, both of whom responded very nicely in terms of bringing down the target protein, in this case hepatitis B surface antigen. But in one of those groups, this was fully half of the group, or nearly half the group, I should say, there was a sustained off-treatment response, which was quite remarkable. So upwards of a year after the last dose was given, we still observed nearly 2 log reductions in surface antigen that were sustained. We saw this as true for other proteins, including hepatitis B E antigen shown here, hepatitis B core-related antigen, and even HBV RNA levels, all behaving in the same kind of phenotypic manner, with one population of patients experiencing a really sustained knockdown of these readouts, prompting the question, is this in fact a successful immune response that's been unleashed to actually bring about a new equilibrium state, or is this a somehow prolonged pharmacodynamic effect of the RNAi itself? And I think further studies need to be done to help sort out between these possibilities. This is another example of another RNAi known as RG6346, which is targeted against hepatitis B surface antigen. And it too, as you can see here, at a variety of different doses compared to placebo, which was flat, brought about, again, about 1.5 to 2 log reductions in HBV surface antigen levels. Again, this is a 4-dose strategy, again, given about every 4 weeks. And again, you see here this rather sustained suppression of surface antigen levels upwards of a year out from the last delivery of RNAi. And in both of the cases of these RNAis, and a number of others that I'm not sharing with you, these were safe and well-tolerated agents, and each seemed to produce about 1.5 to 2 log declines in surface antigen, and these rather sustained suppressions of those protein levels. Again, creating, I think, a very exciting prospect for the possibility of enhancing or likely to developing functional cure. Yet another strategy to knock down viral proteins is a related one. This is using antisense oligonucleotides. These are actually DNAs rather than RNAis, and they too bind HBV RNA species, but the degradation doesn't occur through the RISC complex and through Argonaute proteins like AGO2, but through a ribonuclease called RNase H, shown here. And so again, you can see here in this 6-dose schedule shown here, given in over a 4-week period, these patients, whether on nukes or naive to nukes, experienced rather profound declines in surface antigen levels, as shown in this slide. And really strikingly, for a number of these patients, you can see here the surface antigen levels in red declining, but in ALT levels rising in the context of the decline in surface antigen, raising the very distinct possibility that these ALT flares are related to the very same immune restoration phenomenon that I just described for you a moment ago. And the peak ALTs tend to be higher in those who had more robust reductions in surface antigen. So, so this is a very intriguing set of studies suggesting that, in fact, we are seeing that double-barreled mechanism of action taking place with this strategy of knockdown of viral protein. So moving further down the road, one can get to the step of encapsidation. And here, of course, would be the strategy of blocking the capsid protein itself using a strategy known as capsid assembly modulation. First, a word about core protein. Core is essential for HBV genome packaging and assembly of the virus. It is also essential for hepatitis B DNA replication, and important, as it turns out, for recycling of those nucleocapsids back to the nucleus, and for maintaining cccDNA. So again, there may be multiple mechanisms of action at work here, given the core protein as a multifunctional protein. And so modulators of the capsid, including these proteins or small molecules known as CAMs, or capsid assembly modulators, bring about allosteric modification of the core protein structure, and thereby alter its subsequent ability to assemble the virus, either in an aberrant manner or to just lead to simply empty capsids themselves. And theoretically, if these bind tightly enough and get to enough core protein, they could potentially modulate the synthesis of cccDNA, given what we know about the function of core. And could contribute to greater functional cure rates by working through multiple pathways. So there are a number of capsid assembly modulators in trials, and certainly, again, would appear to be attractive for combination with other classes. And I want to just give you an example of one of them here, called J&J 379, which is given orally, in this case for 24 weeks, in either those who are not on nucleoside analogs, in which there's nearly a 3-log reduction in HBV DNA, or HBV RNA levels shown here, and in those who are already nuke suppressed, there is yet additional effect, about a log additional effect, in those persons who are already suppressed on nukes. In theory, there should be blockade of encapsidation, as we've discussed, and that would lead to a reduction in HBV RNA levels, which is shown here again on the left panel. And as well as a decrease in virus formation, and that's reflected in HBV DNA levels, which I'm not showing here, but were also profoundly affected. Interestingly enough, though, one again might have predicted here reductions in cccDNA and transcription and translation off the cccDNA, but in this instance, we did not see meaningful effects on either surface antigen levels or e antigen levels. And so it may very well be that higher doses or extended durations may be required for disruption of these capsids to prevent this step of the life cycle of cccDNA replenishment. However, we have also seen that when these agents are used as monotherapy, that there may be an increased risk for development of resistance. So these CAMs will likely need to be used in conjunction with other classes of therapy, much as we saw with hepatitis C related agents. And so with that, now that several classes have been developed, we are now starting to see the effects of combination strategies. And so this is an example of combining RNAi plus a CAM plus a nucleoside analog in a small number of patients, all Asian patients, who had a variety of forms of Hep B, E antigen negative and E antigen positive disease. The RNAi 3989 was given as 3 doses, separated by 4 weeks. And then the CAM was given orally for 12 weeks. And you can see that shown here. And what you can see, and these patients were already on entecavir tenofovir therapy and dosed to beyond the end of CAM treatment. And what you can see here is whether through arithmetic or logarithmic scale, there was a profound reduction in hepatitis B surface antigen levels, again, about 1.5 to 2 logs shown here after dosing with this combination strategy. HBV DNA levels shown here on the right profoundly responded to undetectability in the vast majority of patients, really demonstrating that this is a rapid decline in both replication as well as in protein production. And so we're going to really look forward to long-term follow-up data on these patients to determine whether we see functional cure rates develop in excess of the very low numbers that would be expected in any of these patients using standard treatments. So we very much look forward to the results of this and other strategies like it. And then finally, in terms of direct-acting antiviral approaches, there is the step of viral secretion, now or hepatitis B surface antigen secretion in this case. As I said to you, surface antigen is produced in excess and secreted independently as a viral subviral particle known as a surface antigen. And so some strategies have been developed, including one known as the nucleic acid polymer, which has been shown to interfere with the release of these subviral particles or surface antigen, and would be expected to lead to decreased surface antigen secretion, measured, of course, as lower levels in the circulation. Again, as we talked about earlier, this might be expected potentially to enhance immune responses, given surface antigen's role in possibly suppressing or subverting the host immune response, but would not be expected to have any direct antiviral effect on replication itself. So DNAs would not be expected to be altered. So these are data from a nucleic acid polymer known as Rep2139. Again, posited to block this release of surface antigen or subviral particles, and possibly restore immune responses. And so what you'll see here, it's a rather complicated study design, but basically, patients were given tenofovir monotherapy at baseline for a period of time, about 24 weeks. Then the NAP was introduced with PEG interferon on top of the tenofovir versus the addition of PEG interferon only. So the difference here was evaluating NAP plus PEG versus PEG on top of the tenofovir. And what you'll see here in the green bars are those patients receiving the NAPs experiencing a substantial decline in surface antigen in the circulation. A number of weeks later, in this adaptive control group, that group then received NAPs plus tenofovir plus PEG interferon, and in that group we saw again a staggered reduction again of surface antigen. Correspondingly, there was very interesting a ALT rise, as shown here in the green, in each of these particular groups that received NAPs. And most importantly, one can see here on the far right, loss of surface antigen at each of these time points, marching down the line here, of 60% at the end of treatment and about 50% at follow-up weeks 24 and 48. Correspondingly, there was anti-HBs concentrations that were detectable in a similar number of patients, suggesting that these patients were demonstrating features of full-blown S antigen seroconversion, or clear-cut evidence here that supports functional cure. So there appears to be a marked and seemingly durable loss of surface antigen and gain of anti-HBs, but we don't know yet, and further studies are warranted, is whether there are any long-term deleterious effects of intracellular retention of all the surface antigen that's being synthesized, and whether this produces any adverse outcomes. And so we are very excited about learning more about this strategy as well. So that's a little tour of the possible direct acting antiviral approaches. But as we talked about, it's the immune response that is really responsible for producing functional cure in natural resolved infection. DAA strategies may, in and of themselves, be insufficient, as promising as they are, for achieving that functional cure because of the inability potentially to clear latently infected cells. And so here the T cell response, a vigorous and multi-specific T cell response, appears to be the hallmark of successful spontaneous resolution. And can we reinvigorate these responses in chronic infection? And the two arms, of course, of that response are both innate and adaptive immunity. And so innate response, of course, we can talk about cytokine therapy. We already have an approved agent in the form of interferon, but we may be able to more selectively tweak the endogenous interferon system by using toll-like receptor agonists, and perhaps spare ourselves a little bit of that systemic toxicity. And then of course, targeting the T cell itself, or the adaptive immune response, through either T cell vaccine approaches or therapeutic vaccines, or promisingly and interestingly, the use of checkpoint inhibitors directed against exhaustion markers like PD-1 and PD-L1. And again, the idea would be to try to induce flares that clean up those latent reservoirs of infection. So innate responses. So here again, hepatitis B as a pathogen has a variety of nucleic acid signals that can trigger recognition by pattern recognition receptors like toll-like receptors such as 7 and 8. which then induce endogenous interferon synthesis, which can clear out infected hepatocytes. But so too can that interferon downstream trigger other cell types, including NK cells, dendritic cells, to enhance their antigen presentation and cytokine and chemokine production, to in turn trigger adaptive or T cell responses themselves. So innate responses have some antiviral activity, but in turn can trigger those downstream T cell responses. And here's an example of an innate therapeutic approach using a TLR8 agonist known as GS9688 in patients who are already on suppressed on nucleoside analogs. And these patients, whether they be e antigen positive or negative, were randomized to receive either placebo or 2 different doses of GS9688 for a 24-week period, with the idea that the primary endpoint would be a log or greater of hepatitis B surface antigen reductions. So the results of this approach were modest. Surface antigen loss was actually seen in 2 e antigen negative patients, and e antigen loss was seen in 1 e antigen positive patient at the week 24 time point. Moreover, there were, you can see here that if you looked at the group that received treatment, these first 2 groups here in color, there was a more substantial reduction in surface antigen levels, albeit modest, compared to those receiving placebo. Moreover, there was induction of predicted cytokines by those who received GS9688, shown here in the colored bars, such as IL-12, IL-1, RA, and interferon gamma, as compared to the placebo group. So these are promising proof of concept data that support the idea that tweaking of the innate immune response can potentially lead to modest antiviral effects against hepatitis B. Perhaps a more vigorous and robust approach would be targeting the T cell response itself, given the centrality of T cells in controlling HBV and ultimately clearing HBV infection. To date, therapeutic vaccine approaches against hepatitis B have been disappointing. And it may very well be that those T cells are just so exhausted as to simply not be resuscitatable. And so in this regard, it's worth noting that chronic hepatitis B is associated with very high levels on those T cells of PD-1, an exhaustion marker that is associated oftentimes with increased PD-L1, or the ligand of PD-1 in antigen-presenting cells like hepatocytes. And so this combination of engagement of PD-1 by PD-L1 leads in turn to diminished proliferation, cytokine release, and cytotoxicity, i.e. an exhausted functional phenotype of the T cell. And so the premise here would be that blockade of PD-1 or PD-L1 could theoretically de-repress or liberate or rejuvenate that T cell response. And so given the exhausted phenotype of HBV-specific T cells, this may be an appealing target. In fact, this has been tested in Hep C patients, where exhaustion is also seen. A number of years ago, 45 patients before DAAs received anti-PD-1. It was well tolerated. And interestingly enough, at least as a proof of concept, 1 patient experienced a sustained response in that context in association with a significant ALT flare. So can we apply the same approach to hepatitis B? Of course, with the caveat that we may be triggering autoimmunity or immunity against host target proteins and potentially significant disease flares in patients who already have advanced chronic disease. So appropriate caution needs to be planned with these forms of treatment. Well, we do have some lessons learned from a checkpoint inhibitor that was given to patients for hepatocellular carcinoma, some of whom actually harbored hepatitis B. This is Nivolumab, administered to nearly 50 patients with advanced HCC who had failed kinase inhibitors like sorafenib. So in this trial, 11 of these 47 patients were chronically Hep B infected. These patients were predominantly cirrhotic, but with good performance status and compensated cirrhosis with normal Child-Pugh scores. And these patients got a lot of Nivolumab, doses shown here every 2 weeks for, in some patients, up to 2 years. So a lot of checkpoint inhibitor. But interestingly enough, no significant safety concerns. These patients were suppressed for Hep B on oral nukes, and no significant clinical flares or premature discontinuation. There was about a 10% grade 3 ALT rise noted, or 10% of patients had grade 3 ALTs. And there was, but interestingly enough, there was a minimal to modest anti-HBV effect in these cirrhotic patients with HCC. 3 patients of these 11 experienced 1 log surface antigen drop. No one lost surface antigen permanently, but this certainly encouraged us to understand that this was not this was well tolerated and not toxic, and if anything had a modest anti-HBV effect. And so this was then translated into specific patients with Hep B, and this was a trial from New Zealand in which a single very low dose of Nivolumab, 0.3 mg per kg, were administered with a candidate therapeutic vaccine or without that vaccine in chronically HBV mono-infected patients. And this was about 20 such patients who received this. And you can see here that this was the measured hepatitis B surface antigen levels. And you can see again that those patients experienced modest reductions in their surface antigen, but notably, 1 patient in the Nivo monotherapy single dose group experienced a profound reduction. In fact, loss of surface antigen, as shown here in the blue line in this single patient profile, together with an ALT flare shown in red, and the acquisition of a more brisk HBV-specific T cell response. So functional cure was accomplished in 1 of these patients with a corresponding improvement in HBV-specific immunity. And this patient 8 months later remained surface antigen negative with anti-HBs seroconversion. And again, minimal toxicity, 1 grade 3 ALT elevation, and everyone else tolerated this single dose well. So additional trials are planned with important translational components to understand the correlates of the receipt of anti-PD-1 in other patients with chronic hepatitis B mono-infection. So a very exciting approach being tested. So as we think about possible pathways to achievement of functional cure of hepatitis B, we are really thinking about 3 real major classes of treatment. Those that block replication itself, such as best typified by the nukes, but also include the capsid assembly modulators, RNAis, entry inhibitors, and even cccDNA inhibitors. Those that lower viral protein or antigen burden, RNAi again being the showcase here, along with antisense, nucleic acid polymers, and cccDNA inhibitors. And then boosting of the immune response itself through, again, the demonstration project here with the anti-PD-1, but also TLR agonist, therapeutic vaccines, and possibly even T cell reprogramming. And it is through this potential multipartite approach that we could potentially envision a high or a much more impressive functional cure rate strategy moving forward in terms of getting those patients to surface antigen loss. So I hope I've convinced you that HBV cure is a much more elusive target compared to Hep C due to its highly stable latent form and what we understand about the viral life cycle. That nuke therapy is a successful suppressive but an inadequate cure strategy. But that functional cure is a realistic goal, keeping in mind that we are trying to reproduce the playbook that nature has given us with spontaneous resolution of patients who acquire HBV in adulthood. Fundamentally, new approaches to Hep B are under study with multiple representatives in multiple classes, including RNAi, antisense, CAMs, NAPs, and immune modifiers, each of which show promise as individual classes and hopefully even more so in combination, and which we believe will be essential for achievement of higher rates of functional and, for that matter, sterilizing cure. And so we think that DAAs and immune modifiers will be very important parts of those combinations, with the intriguing question about whether RNAi alone, given some of the prolonged surface antigen reduction or even loss data, could such an approach alone suffice in a subset of patients. But these are exciting times for Hep B because as a group and a field, we've pivoted and now taken a look at everything under the hood. And to consider not just the obvious target, but many other targets that are rational. So with that, I'll thank you and thank you for your attention and thank you for the honor and privilege of speaking with you all today.